Welcome to this week's episode of Brainstorm, where we give you a glimpse into the world of science for Wednesday, February 6th, 2013. We begin with a story from the world of agriculture, with an update from our favorite field of research, genetically engineered crops. And this isn't even scary engineering like building in pesticides or changing the nutritional content of a plant. This research, done by Cornell, is about straight-up increasing the efficiency of plants. You see, there are two main types of photosynthesis that plants can perform. A lot of plants, including many crops, perform C3 photosynthesis, which is less efficient. C4 photosynthesis evolved much later in around 60 plants independently, including grass, sugarcane, and maize. The difference relates to the actual anatomy of the leaves and allows C4 plants to handle droughts better and require less nitrogen. In all plants, an enzyme called rubisco is responsible for capturing carbon dioxide for use in photosynthesis. But in C3 plants, the enzyme also interacts with oxygen, reducing the overall efficiency by 30 to 40%. Whereas C4 plants perform this process in two steps. First in the additional outer leaf layer called the mesophyll, and then the carbon dioxide is brought to the bundle sheath cells around the leaf veins. Only the second step involves the enzyme rubisco, and inside cells with very low amounts of oxygen. Because these bundle sheath cells looked like certain cells inside the roots, researchers examined the genes controlling both of them. Working with strains of maize, they identified a gene that is a key regulator in C4 leaf anatomy, dubbed scarecrow. It'll be some time before new C4 crops like wheat and rice are engineered, but it's an important first step, one that could lead to a 50% increase in yields, even in harsher conditions. Next is an update from the world of biotechnology. A group from the University of Minnesota have found a way of culturing a tricky and potentially useful bacteria. Particularly, iron-oxidizing bacteria which use dissolved iron as a primary component in their respiration. Now, these sorts of bacteria can be found all over the place, usually where a low-oxygen environment interacts with a high-oxygen environment. Sometimes they're beneficial, helping iron cycle through an environment, or damaging, adding to the corrosion of steel pipes due to a byproduct of their metabolism being rust. Because of all this, they've been extremely difficult to culture in a lab. Since scientists think the actual oxidation step happens on the cell membrane, a simple solution was discovered: feeding the bacteria straight electrons. First, it was just attempting to grow the bacteria on an electrode with a growth medium, but eventually they removed the iron from the medium and demonstrated the bacteria could grow with just electricity. This is important not only for research, but it could have some serious practical applications. Since these bacteria can live off essentially just electrons, they could be made into a kind of reverse bioreactor, converting electricity from sources like wind and solar into biological molecules to be used as fuel. Although such energy storage technology is far off, this is an important first step. Finally, we have news from the world of medicine. Every intelligent person in the Brainstorm audience will agree that vaccines are pretty awesome, but they have their limitations. Conventionally, they're made from dead or weakened versions of a virus that our body then reacts to. This isn't necessarily easy to manufacture, store, or transport. And for diseases like HIV, injecting even disabled viruses could be risky. In these vaccines, the body is mostly reacting to the proteins in the virus's outer shell. However, straight viral DNA has been shown to impart a similar immunity in mice. The problem is replicating this result in humans. One attempt involved injecting the DNA just under the skin and applying an electrical field. Called electroporation, it created temporary holes in the skin cells, allowing viral DNA to get in. Results were inconsistent, and as you may have guessed, somewhat painful. Fortunately, MIT scientists think they may have solved the issue with the help of material science. They designed a vaccine patch, consisting of multiple layers of biodegradable polymer embedded with the target DNA. Microneedles painlessly inject the DNA over a period of days or weeks as the polymer layers degrade from contact with moisture. Both the slow release and the mixing of the polymer and DNA prevents the body from breaking down the viral DNA too quickly. In addition, virus-like RNA is added to trigger some inflammation and bring more immune cells to the site of the patch. Tests involving simian HIV and macaque skin cultures are promising. If further testing is successful, this could lead to effective, stable, and painless vaccines for virtually any virus. Well, hope you enjoyed this episode. If you did, please consider subscribing and be sure to check the links in the video description.